can start the class. Please take out your PDF. We have already sent the PDF, so please take it out. So what we are going to do today is, since we have already given the notes on this chapter, we are just going to discuss and to see as per what is important from examination point of view. We don't have to write anything because you have your notes already, right? So talking about this new chapter, plant growth and hormones or plant growth and development, what are the questions that are generally asked from this part? The questions generally are asked from the hormone section, right? So in this chapter, we are going to study certain hormones, or we are talking about phytohormones. And whenever we talk about phytohormones, we'll be talking about 5 of them: orphan, gibberellin, cytokinin, hyaline, and acetic acid. So what is the physiological role of these hormones, right? Where they are synthesized. We have something called as the bioassay. We have a word called as the precursor of the hormone, etc. etc. So under these points, we are going to study the hormone section. Apart from the hormone section, the first part of the lesson talks about what? General growth. Growth what are the different types of growth that we see? In case of plants, the growth is different. In case of animals, the growth is different. Plus certain types of growths like relative growth, absolute growth, right? Next, arithmetic growth, geometric growth. So the first part talks about growth in general. The second part talks about the phytohormones and the role of the phytohormones in growth and development of a plant, right? So we just start the lesson. Talking about the first page of your PDF, first up what is growth? So what is growth we understand, right? It's a permanent irreversible process or reaction. And if you are saying that the growth has increased, so what we understand, increase in fresh weight, dry weight, size, length, height, etc. The growth has increased, right? Next is, we have 2 words, one is called as quiescent and the other is called as dormancy, right? Out of these 2 words, I think dormancy is a word that you have come across, that's very common, no? If I say a particular plant part is being dormant, what we understand by that statement? Eta part dormant progressed into one, or if you are saying that a seed is dormant, maybe the germination has not taken place. So that is the time we use the word dormant, right? But together with the word dormancy, we use a word that is called as quiescence also, right? So quiescence and dormancy, both of them are suspension of growth. Suspension of growth means the growth does not take place at that part, right? But if the suspension of growth is due to exogenous factors, so a suspension of growth means the growth does not take place, right? So why doesn't the growth take place? Due to certain reason, right? If the growth does not take place because of some outward features or some environmental conditions or some conditions outside the body, that is called as exogenous, and at that time we will be using the word called as quiescence. But if the dormancy is because of some internal factors of the plant, that is the time we use the word called as dormancy. So duita mane ki suspension of growth. Baki factors of then factors of So just know the word, that's all. As far as animals and plants are concerned, animals we have a growth that is called as the diffuse growth, and plant we have localized growth. Localized growth means they grow only in particular areas, that is what is called as a localized growth. And whenever we talk about growth in case of plants, generally maximum growth has been restricted to the root apex and the shoot apex. So we have a local area, we have a particular area where the growth continues throughout the entire lifetime. But if you are talking about animals, animals do not grow throughout their lifetime. Up to a certain amount of time, the growth continues, and after that the growth gradually stops, right? Another words, limited growth and unlimited growth, just go through the PDF. In plants, the growth could be limited or it could be unlimited. Limited is also called as determinate and unlimited is called as indeterminate. I think this part is understood, right? So growth or actually information we have. Next time development. What is development? All the changes that you can see in the body of an organism, that is what is called as development. Right? Next, tinta terms. These 3 terms we have been speaking about it in so many cases. Differentiation, redifferentiation and redifferentiation, right? Differentiation means whenever a cell is matured enough to take up the function, that is what is called as differentiation. And other way around, what we know, that the meristematic cells becomes permanent and they do not divide again, and that is what is called as differentiation, right? Now each and every cell, let's start start from one single cell and the cell that divides is called as meristematic. Meristematic cell will divide key form a group of cells, and these group of cells at a particular time they will stop division and then they will become permanent. যেতিয়া permanent হৈ যায়, meristematic cells will actually permanent হৈ যায়. At that time we use the word key that the cells are mature, the cells are differentiated, etc. Right? So the point to be remembered is differentiated cells do not divide. Differentiated cells like permanent cells are always differentiated. But sometimes what we see, differentiated cells also become meristematic. In normal cases, meristematic cell becomes permanent, and once we are using the word permanent, they will not divide. But there are certain examples, there are certain exceptions where a permanent cell divide again, and at that time we use the word called as dedifferentiation. Right? So permanent cell asile ki ako divide korile tamane ki ako meristematic hoy gol teti ami process to ki become dedifferentiation. Ako meristematic hoy joa cell to ako mature hobo lagibo বুজি পাইছা না এটা cell ase meristematic cell to hole convert hole permanent hole once it got converted to permanent it is mature, it will not divide. But sometimes a permanent cell may become meristematic again, right? At that time that process permanent into meristematic, that process is called as the dedifferentiation, right? So permanent ki hobol meristematic hobol, that newly formed meristematic cell will also become mature at a particular time, will again become permanent, right? At that time we use a word called as redifferentiated. Just go through the three words and see whether you have understood or not. Differentiated, dedifferentiated and redifferentiated. Differentiated ki hobo, so we have a meristematic cell ki hole convert hoise permanent hole that process to ami ki korso differentiation. Sometimes what happens, permanent cell again becomes meristematic and this is called as ki dedifferentiation. So permanent cell becomes meristematic. This meristematic cell again has to become permanent, and this process would be called the, I think it's clear now, right? Whenever we talk about growth, we generally have to talk about all this. And redifferentiation, everything generally associated with what? One common example, no? We will have to talk about that secondary growth in case of plants. Because we know in case of secondary growth, cambium divides, no? Cambium itself is a permanent cell, it should not have divided. But we know that certain permanent cells also divide. One example is cambium. And once the cambium divide, it leads to the formation of the secondary growth in case of plants. Right? Okay. Next, we have another word that is called as plasticity. What is plasticity? The ability to change according to the internal or external stimuli. That is what is called as plasticity. Every organism, due to certain reason, if it changes its morphology, if it changes certain types of anatomy, that is what is called as plasticity. Generally, we are concerned with the morphological changes of an organism. Morphological change maybe because of some external features or maybe because of some internal factors, right? So this change is what is called as plasticity. And whenever we talk about plasticity, intrinsic plasticity and extrinsic. Intrinsic obviously internal factors, or extrinsic external factors. We have certain examples. Plasticity basically 8 example monoretically. First is coriander, second is ranunculus. Right? Ranunculus, I think you all remember when we were talking about it in morphology, we have talked about it. Ranunculus, it's a water plant, right? It shows what? The same plant shows 2 different types of leaves. We have a phenomenon called as heterophilia, right? This heterophilia nature is because of what? Because of plasticity or the changes due to the environment, right? The plant is called as ranunculus. It will have certain leaves above the water and certain leaves below the water. Above the water, leaves they are lobed and they are broad. Below the water, the leaves of the plant ranunculus, leaves dissected leaves, right? So according to where the leaves are present, above water and below water, the leaves have shown modification, and this is what is called as plasticity. This plant has 2 different types of leaves, and the phenomenon is called as the heterophilia. Clear? Okay. Next is factors affecting plant growth. This is something very common, no? What are the factors that affect plant growth? Nothing to read about that. Now, how will you measure growth? Measure growth, you can measure in terms of number of cells. Heterophilia. Heterophilia, hetero is different, phily is leaves. One single plant having two different type of morphological leaves is called as heterophilia. Right? One best example that we have seen in your regular life is coriander. In case of coriander, I think you know what coriander is, right? So when the coriander is young, the leaves are different. Once it starts or approaches flowering, the leaves changes. Right? It becomes dissected then. Normally or lobe or something, right? So that is what is called as heterophilia. Same plant having two different types of leaves. Next is measurement of growth. How will you measure? You can measure growth in terms of fresh weight, dry weight, length, volume, number, surface, etc. No? Adult plant grow korise boli kole, what is the most common way that we see or we observe is the increase in height. No, grow korise mane ki hai? Height is and suppose we are talking about unicellular organisms, and if we say our unicellular organism is growing, what we understand? The number of cells is increased, right? It already korise ami Next is phases of growth. When we talk about growth, if I say our organism is growing mane ki tinta bostu hobo. First, the cells will divide. The divided cell should elongate and ultimately it should become mature. So a cell is growing means what? The cell is dividing. After division, what happens? Now, whenever a cell divides, it does not mean that it will keep on dividing its entire lifetime. At a time it will be stable or permanent hoboi lagibo. তেতিয়া কি ফাংশনস বিলাক কৰিব তো সেলটোৱে ডিভাইড কৰাৰ পিছত কি কৰিব elongate কৰিব and then it will become mature and do the function. These three phases collectively say that the growth has taken place in a cell, right? As far as elongation is concerned, cell division we have already done, so we know, right? Mitosis hobo pare, meiosis hobo pare, no? As far as the next phase is concerned, that is elongation. Who is responsible for elongation of a cell? It is the vacuole that is responsible, and the process is called as vacuolization, right? Vacuole to get it responsible because we know whenever the absorption of water and minerals takes place in a cell, vacuole kole jaibo, vacuole jaibo, and the vacuole when it stores, it starts storing, it will enlarge. Once it enlarges, the protoplasm is pushed to the periphery and ultimately it enlarges, and this is what is called as cell elongation. Once the process is done, it becomes mature and do its function. Next is talking about growth in case of organisms. If we draw it in terms of a graph or we study it in terms of a graph, what is the type of a graph that we see? We see a sigmoid curve, right? What is sigmoid curve? We have already done. Organism and population of J shape, S shape we have done. What we are talking about here is the S-shaped graph. No? Whenever we talk about S-shape, what we understand that there are certain phases, no? Lag phase, log phase, and the steady phase. Right? When we talk about the lag phase, what we remember, the growth is very less. It has just started. Log phase. That is lag phase, exponential growth. बहुत basically grow करेगा. बहुत basically grow करार पिसर की हो steady हो जाएगा, and that is what is called as the stationary phase, and gradually senescence. Right? Okay, now we are talking about growth. Now we know if I say a cell is growing, we understand the cell is dividing, the cell is elongating, and the cell is getting mature, right? How is it growing? How much is it growing? What is the rate, right? If you want to study something like this, we have to talk about growth rates. And whenever we use the word rate, what do we consider? We consider the time factor also. যে এটা টাইমত কিমান গ্রো কৰিছে এই টাইম ফ্যাক্টৰটো যদি আমি কনসিডাৰ কৰো তেতিয়া আমি কি ওয়ার্ড ইউজ কৰো ৰেট ৰাইট এনেটো নরমালি আমি কি ইউজ কৰো গ্রোথ ৰাইট কিন্তু গ্রোথ পার টাইম is something called as rate, right? So there are three types, that is the arithmetic growth, geometric growth, relative and absolute growth. Generally you can talk about it as 4 types or you can say it as 2 types also. কিন্তু এইটা গ্রোথ আমি জানিব লাগিব arithmetic growth, geometric growth. That is 4 types of growth could be seen. Either it could be arithmetic or it could be geometric, it could be relative, it could be absolute. Depending on which part of the plant you are talking about, right? That part will either show arithmetic growth or it will show geometric growth, right? Part to part in a plant of different types of growth dekha pare. Coming to the first one, arithmetic, no? Arithmetic growth and geometric growth. What is the basic difference? Growth fully what we understand, we have a cell. The cell will divide and ultimately form 2 cells. This is growth, right? Now what I am saying is growth could be of 2 types. One is arithmetic growth. Arithmetic growth of key hoy. Then the first cell divides into two cells. What next? A2 cell divide korbo laise, A2 cell divide korbo laise. Kintu ki hoy je the first cell divides into two cells. Out of this, one cell does not divide and instead it becomes big, enlarged or mature visible, and the other cell will divide and form two cells. Again the same process. Out of the two cells, maybe one cell will not divide, it will become enlarged. The other cell will divide and form two cells. So if this is the thing, this type of a growth is called as arithmetic growth. Whereas on the other hand, if we are talking about the geometric growth, the normal type of growth that we always speak about. Each and every cell will divide. We call geometric growth. Right? অকল এটা সেল ডিভাইড কৰিব প্ৰত্যেকবাৰে কি হ'ব এটা সেল ডিভাইড নকৰে মেচিউৰ হৈ যাব কাৰণ obviously if we are talking about an organism, if we are talking about a plant, if we are talking about a human being, any type of organism, obviously life starts from a single cell, right? That cell cannot be dividing and dividing and dividing, and it cannot form only a collection of cells. At a particular time the growth needs to get stopped, right? It has to get mature. So we see both arithmetic growth and geometric growth depending on which plant part we are talking about, right? So could you figure out the difference between the two? Both the cells divide, geometric growth. Only one cell divide, that is called as arithmetic growth. Whenever we talk about arithmetic growth, another diagram that is given in your NCERT book, no? Another diagram here. This is the cell. Next time what will happen? This will form two cells, right? Out of these two cells, one will not divide, right? And the other will divide. Next step, what we will see? The original A2 acylate. This cell will divide into. This cell will not divide. Ultimately, this will divide. Finally, again, if you are talking about the next step, what will I say? Original acylate, A2 acylate. This cell will divide into two. Out of this two, one will not divide and one will divide. As a result, this layer of cells will keep on increasing and getting mature. Here. Okay, just go through the period and the notes that is given. Rate of growth is constant. One daughter cell remains meristematic while the other grows and differentiates. Increase in growth occurs in arithmetic progression. It is found in root and shoot effects. Arithmetic growth is expressed in, you have a formula to calculate the arithmetic growth, you have a formula to calculate the geometric growth also. From botany point of view, calculations or mathematical calculations regarding these has never been asked in your NEET, right? For this question, they identified the formula used to calculate arithmetic growth. It is only the formula that you have to remember, right? So arithmetic growth formula to what rakhiba? LT is equal to LO plus RT. And if you are talking about LT and there is a graph, no? Graph analysis, right? 18% social PDF or PDC. LO, what is LO? The initial growth, no? Because we know if you are talking about any organism, because of best example, human beings, jeta baby to ahe no, baby to jeta deliver hoy agote growth hoy ahe na. Mother's body bhitor to already grow hoy goise. Tar pisote ami je kon the baby has attained 1 year. 1 year means what? 1 year after delivery. That does not mean he has just started growing at that point where he has been delivered, no? So that point is what is this is called as initial. Initial is a hakibo tar pori monthly growth is that can be arithmetic growth. So what is important? It is seen in both root and shoot effects, that is important. Second is it is expressed in the formula, so formula is important. And the curve. Is this clear? Next is we come to geometric growth. Geometric growth we understood each and every cell will divide. The most common type of growth that we talk about. Normally geometric growth को take about in case of unicellular organisms where each and every cell will keep on dividing, right? Okay. Common in unicellular organisms, every cell divides. Growth. This type of a growth is seen in all cells, tissues, organs, developing seeds, germinating seeds, etc. In case of embryo, initially shows geometric growth, but later arithmetic growth. This is a point that you need to understand. We are talking about the embryo. As we have already discussed, what we said is depending on which part of the plant you are talking about, different types of growth will be observed, right? If you are talking about the root and the shoot apex, the type of growth that we see is generally arithmetic. Remaining organs, when we talk about organs increasing in size, increasing in volume to increase number of cells generally we talk about geometric growth, right? But we have written something about the embryo. Embryo ki hoy je n into n will give you the zygote. The zygote is a single cell. This cell will divide and re-divide, right? And ultimately will form a collection of cells, and that collection of cells is called as embryo, right? So we understand that we had a cell. Eto kami ki koiso? Zygote. A zygote ke duta form korle, sarita form korle, eneke form kore pele ki form korle? Embryo. Now, this process cannot keep on going like this. Onward embryo is a form koribo, etu to nohoy embryo par to baby plant to form koribo libo. Hoy na. So yar pora jodi intake collection khonar pora jodi baby plant form koribo lage, radical form koribo lage, pleural form koribo lage, then some cells obviously has to become mature, right? If you are talking about being mature, which type of a growth? Yar is of ki hobo arithmetic growth will progress. Etu pora etu le ki hobo geometric growth. Is it understood? यानी कि वो केटामल cells है वो केटामल लाहे mature और radical and pleural form होगा. So embryo, what we can say is we can take the example of an organ where initially it shows arithmetic growth. Initially it shows geometric growth, and later it converts itself into arithmetic growth. Arithmetic growth किहो करने को बोला जाता है कि cells बिला कॉमन के mature तो बिला function करे. खाली cell बिला divide कोई numbers बिला जिन्हें increase कोई बोला के जितना ले किलो का growth होगा geometric. If the cells seem to get mature gradually, at one time the cells does not get mature, no? Gradually at that time mature to a collection form korbo. At that time, what is the type of growth rate and growth we see? This one? Okay. We know the zygote, right? Zygote mane ki hoy? N into N will give you a zygote. Male gamete, female gamete fuse korbo eta cell form kore. Ei cell to ki hoy zygote. Zygote ki hole convert hoy embryo. Embryo ki form kore baby plant. Radical pleural tartar to hoy. So ei eta cell or embryo form koribo lagile collection of cells hobo lagibo na lagibo. So eta cell or collection ke liye hobo divide koribo lagibo, right? So etu cell or prothome duta form korile duta or saita form korile saita or aata form korile. So ei je protectu cell is divide kori bari goi ase tata ki boli kom geometric hol. Etia embryo to onobor enike dangor hol to goi thakibo nware. Eta homoy to be stable hobo lagibo, mature hobo lagibo, karon tar pora to plantal ulabo lagibo no. Plantal ulabo lagile ei process to stop hobo lagibo na lage. Lagibo lagi pele eta duta cell ki mature hol pele konobai radical to form koribo, konobai pleural to form koribo. So tetia ki growth hobo tetia arithmetic. At the time of the formation of the baby plant ki hobo arithmetic growth. Karon ei duti collection of ketamon cells mature hol, it has to function as a radical, it has to function as a pleural. Right? So mature hobole generally আমি কি use করি arithmetic growth, collection বা number of cells বঢ়াবলে আমি কি use করি geometric growth. Yeah. Next again, geometric growth is calculated by the formula. Right? खाली formula to जानी बारे को लिखो. So since we are talking about growth rate, first two growth rate that we have discussed is arithmetic growth and geometric growth. After this, we have two terms again, that is absolute growth and relative growth. Our absolute growth and relative growth or question Sahib, the diagram is given in the NCERT book also. Right, that leaf, no? The leaf ki asked me. What we are taking here is two leaves, one leaf, smaller leaf, one bigger leaf, right? And both the leaves are growing, the dotted line shows the growth, right? So if this leaf is also growing, ekini kiman growth korise? maybe 5 centimeter. Okay, again this is a bigger leaf, it is also growing. Yate kiman grow korise 5 centimeter. Etia duitare no doubt they have grown 5 centimeter each. In terms of relative and absolute, the calculation is different, right? So yate ki hobo the duta type of growth dekhi, eta hol relative growth, eta hol absolute growth. If you talk about relative, but first let's talk about absolute. Absolute growth mane ki eta time of kiman grow korise asile etu leaf grow korise kiman grow korise, maybe time to maybe 7 days, right? So we have taken 2 leaves and observed it for 7 days. Etu leaf 7 dinot 5 cm grow korise, etu leaf 7 dinot 5 cm grow korise. So the amount that is grown per unit time is called as K, absolute growth, right? Growth per unit time is absolute growth. But in case of relative growth, what you have to consider is the initial growth also. Je originally kiman asile, yate amu jodi absolute growth count korbo, absolute growth count korute original growth to kiman asile, this is a factor that we don't consider. If you consider the initial growth also, that type of a growth rate is called as relative. So what is the difference between absolute and relative? Absolute talks about only 2 things, what? The growth and the time. Relative talks about 3 things, the growth, the time, the initial growth. Kiman asile agore 20, yate kiman asile 5. So 5 or 5 cm ase, 20 or 5 cm ase, right? Hetu jodi calculate koro eta taram ki likho relative. Initial dhorile kodai relative, initial jodi nohora eta that is absolute, right? As far as counting is concerned or calculating is concerned, no, just see relative growth calculate koribole eta formula dia ase. So the last line of the PDF, page 40, page not 74. Okay. If you don't consider the initial factor, then it is called as absolute. When you talk about absolute growth, A is also growing, B is also growing, A is growing by 5 cm, B is also growing by 5 cm within a time of 7 days. So absolute growth kiman hobo? Duitare growth K hobo. Kyo hobo? U 5 cm grow korise, U 5 cm grow korise, right? Kintu relative growth jodi sound ইয়াত কি কম ই 5 সেন্টিমিটাৰৰ পৰা 5 সেন্টিমিটাৰ গ্ৰো কৰিছে, ই 20 সেন্টিমিটাৰৰ পৰা 5 সেন্টিমিটাৰ গ্ৰো কৰিছে, হেতু হলে ৰিলেটিভ হৈ যাব, right? And if you don't consider initial, A is also growing by 5, B is also growing by 5, both of them are growing by 5, so they have the same absolute growth, but they have different relative growth, right? Different relative growth. It is leaf A, jodi ami relative growth calculate koribo lage, you have to go by this. So what will be? Yes, original kiman asile 5, initial 5 asile, grow kiman korise 5, right? Into kiman 100. So relative growth of A would be kiman 100%, right? But if you are going to calculate the relative growth of B, 100 hobo san kiman 25, right? Karon jodi B count koro teta ki hobo? Initial kiman asile 20, kiman grow korise 5, right? So obviously 100 no hobo, right? So this is the difference between absolute and relative. In case of absolute, both the leaf has the same absolute growth as 5 cm. If you are talking about relative, smaller leaf through kiman growth basi. Anyway ki ko whenever the body is young, it grows faster, right? Anyway, if you are talking about a baby also, no, if you are talking about a human being, growth phase 2, I mean, if you are comparing 2 situations, one, you are taking a baby 1 year old or maybe 6 months old, and in the other case you are considering a person with maybe 16 years, 15 years. 16 years turned into 17 years, 6 months turned into 1 year. Car growth to visible hobo. 16 years to 17 years, so what is the difference that we are going to see? Nothing or almost something, right? So, Oru postal growth to Hodai DC, initial or Prami count no? Because we consider the initial point. So, we are considering taking into mind that initial factor also, right? So, ultimately, what is the question that is asked? The question that is asked for need is, you have to calculate the relative growth of the leaves. Not necessarily 20 or 5 years, it could be any number. When you know this, you can take out the relative. Just go through that part, whether you have understood or not. Per unit time you can comment on the 7 days. Do it at a time on hand considerable. 8 to 3 days, 8 to 5 days, then you cannot. Just see absolute and relative growth to busy by So what we can conclude, the absolute growth for A and B is the same, that is 5 centimeter, but the relative growth is different. In case of A, it is 25, in case of B, 100. Here. Next we can proceed towards the next part, conditions for growth. There are certain conditions required for growth. Again, GK wise, synthesis of more protoplasm. Obviously, if a cell has more protoplasm, it will grow, right? Next is cell division, cell elongation, cell differentiation we have talked about. The conditions necessary are supply of nutrients. What is the relation between nutrients and growth? Directly proportionate. Nutrients growth directly proportional, right? Water and growth. Water growth, right? Light. We have already studied photosynthesis and light and all those we have talked about. Etiolation is a term also we have discussed, if you remember. Right? Oxygen. Obviously, oxygen growth of aerobic organism. Temperature, carbohydrate. Okay, coming to this point, carbohydrate nitrogen ratio. The first point is important, right? Tactile classes also. In a plant, more nitrogenous compounds as compared to carbohydrate produces more protoplasm. Eta cell grow korile ki lagibo basic protoplasm lagibo. Basic protoplasm ketia hobo? Jete nitrogen basic thakibo, carbohydrate kom thakibo. Right? So, hetu point of question is that more nitrogenous compounds, less carbohydrate results into the synthesis of more protoplasm. Similarly, one point about the next one, less nitrogenous compounds results in synthesis of more wall material. The cell wall will become thicker, right? Growth protoplasm to basic nitrogenous compounds to as compared basic complicated nitrogen wall cell wall will become thick. Right? So up till here we have talked something about the growth, we have talked about development and the different types of growth that we see. Absolute, relative, geometric, arithmetic. Right? Oh. As a whole we meant to say. Cell wall will be cell wall, cell membrane to protoplasm and boundary is specifically cell wall will be synthesis of more wall materials. So wall materials could be, we can talk about the secondary wall, middle level also, right? Secondary wall to over, primary wall to over. Clear? Okay. Next, the concept of photoperiodicity. We have a term called as photoperiod. Photoperiod means the phenomenon is called as photoperiodism. In a photoperiod, period is a time. Photoperiod means time of light, right? Each and every plant will have their own respective requirements for light to flower. This phenomenon is called as photoperiodism. Their requirement of light is called as photoperiod. They take such an amount of photoperiod to do what? To flower, and that process is called as photoperiodism. So is the two words clear? Photoperiod and photoperiodism ki bostu. Photoperiod means time requirement. Day length kiman lage eta plantor, kiman khini sunlight lage, kiman hours of sunlight lage. That is called as photoperiod. 5 hours of sunlight loi pelai moi flower korisu. The process is called as photoperiodism. So the relative length of day and night is called as photoperiod. The response of a plant to the photoperiod expressed in terms of flowering is called as photoperiodism. Is the statement clear? Okay. Question kiya hai, who has discovered? So name of the scientist is important, right? Gardner and Ellard. Experimented on Maryland Memot. That is also important. The concept of photoperiodism. What experiment korisile? Tobacco korisile. Tobacco kuntu variety? Maryland Memot variety of tobacco. Right? Prothamate to kunba eta plant experiment koribo lagibo na. Tetiya phenomenon to api ulabo paribo. So tobacco, or we have the scientific name that is Nicotiana tobacco. So according to Gardner and Ellard, what he said is, on the basis of photoperiod requirements, plants has been classified into 3 types, that is short day plants, long day plants and day neutral plants. Coming to this section, question kiya hai? Identify the short day plant, identify the long day plant. So examples are important, right? Good examples, one example we are taking for example, if you are talking about Gentium question to ahiye hai. Short day plant boli kole, one most important example is Gentium. Okay, coming to these plants, no? Short day plant and long day plant, what do we understand? Short day boli kole ki mon kore. When you have a plant, the plant will require less amount of daylight and more night to flower. That is what is called a short day, right? If I am talking about a long day plant, dinot besi din lagibo. Ba besi sunlight besi ke lagibo, rati factor to kom ke lagibo, teta kami become long day, right? Especially if you are talking about winter plants could be categorized under long day and short day. It is always short day, right? Just going to the short day plants. Short day plants are ki lage? The day length to short length. Next is what is important? Examples. Third point that is very, very important. Short day plants always require uninterrupted dark condition. This is the line understood. Short day plants, suppose I am talking about a plant, right? Right? Suppose either plant or day length मान लगे थोड़ा 5 hours, और night लगे थोड़ा बाकी 4 hours. Or maybe if you are talking about 24 hours, maybe 4 hours of day and 20 hours of night, right? So they will require short day, long light. So a plant का कभी क्या लिखा? SDP. This is the requirement for flowering phenomenon to क्या लिखा? Photoperiodism, right? One important thing is, एटू की होय dark phase. Dark phase नाटक केनिका लगे uninterrupted. Short day plants always want an uninterrupted dark period to flower. Uninterrupted dark period माने कि if suppose within this 20 hours if you expose the plant to light, this plant will not flower. मुझे पैसा दिनों किमान घंटा ऊपर लगे 4 घंटा अंधार किमान लगे 20 घंटा ए अंधारा होवल তুমি যদি আর্টিফিশিয়ালি লাইট দিয়া বা কোনবা এটা বেলেগ জায়গাত লৈ যোৱা যদি কি সানলাইট আছে তেতিয়া হলে এটো ইন্টারঅ্যাক্ট হ'ব যদি ইন্টারঅ্যাক্ট হৈ যায় কি ফ্লাওয়ারিং নকৰে so short day plants are most important requirements ki they always require uninterrupted dark condition. If this dark period is disturbed by a flash of light, then this plant will not flower. It's a point. Okay, just read the lines. Short day plants are lines. Example: gentian very important, viola important, soyabean. Underline korai ase no. important. Just read the 3 lines that are written. কি লাগে? Continuous dark period লাগে. Continuous dark period নাপালে কি হ'ব? Flowering নকৰিব. তাহাঁতক দিনৰ পোহৰ বেছিকে লাগে নে ৰাতি বেছিকে লাগে? ৰাতি বেছিকে লাগে, because so they are called as short day, right? Again, coming to this one, we are talking about a short day. Maybe its requirement is 4 hours of light, 20 hours of dark. If I expose the same plant to 3 hours of day and 21 hours of dark period, what will happen? Are you understanding? That actual critical photoperiod to it is that নিজৰ requirement কিমান? Our নিজৰ requirement was 4 and 20. 4 light, 20 dark. What I do, I take this plant and I decrease the daytime, increase the night time. Then what will happen? Then the flowering will become much earlier. Why? Because ইহঁতক যিমান ৰাতিপুৱা সিমান ভাল. The more dark they are exposed to, the more early the flowering will be. Clear? Are the 3 lines clear? Just see. Prolongation of the continuous dark period initiates early and good flowering in SDB. The last line is what I have explained just now. Right? Last line of SDB, just see. Demand lage activity basically dark period here, it's good for the plant. Which plant? SDB. Right? Next, long day plants. Long day plants, again examples: Burnie, henbane, potato, carrot, junta, mark. So out of this question has been asked about henbane. Henbane or birat question aise? ठीक है? Reddish, burnie. Okay. Again, long day plants, as the name suggests, we understand the meaning. ताहर की लगी बोल दिन और light to basically लगी बोल रात to कम के लगी बोल. A brief exposure of red light in the dark period. Okay. Below the examples, a statement दिया है इसको long day plants or examples or line এটা लिखा है. A brief exposure of red light in the dark period stimulates flowering in LDB. Means what? LDB ki ho? If you are talking about LDB, tahara more ki lage, less ki lage, more light lage, right? Less dark lage. Suppose it has 20 hours, again we come to the same example, 4 hours, right? ইয়াতে 20 নিদি পেলাই যদি মই 21 দিও বা 23 দিও তেতিয়ালে কি হ'ব good for the plant because it requires more light. হেতু কি আছে dark বেছিকে লাগে, so if you increase the darkness, good for the plant. ইয়াতে increase the light, good for the plant. Clear? And thirdly, we have day neutral plants. Day neutral plants, they do not have any relation with the day length and the night length, right? They are neutral. Okay, before discussing any further examples, I think everyone has heard the example of I think you know what a spinach is. Spinach to know? Spinach is an example of long day plant and short day plant. First thing, spinach আমি কেতিয়া পাওঁ? কিতাপৰ কথা বাদ দিয়া, আমি খাবলৈ কেতিয়া পাওঁ? গৰম দিনৰ পাওঁ নে ঠাণ্ডা দিনৰ পাওঁ? ঠাণ্ডা দিনৰ পাওঁ। যদি ঠাণ্ডা দিনৰ পাওঁ কি হ'ব লাগিছিলে? Short day plant, কিন্তু কি short day plant or long day plant? It's a long day plant. Why it is a long day plant? Because the part of the spinach that we eat is not the flowers but the leaves. Photoperiodism is related with not the leaves, it's related with the flowering. Spinach or flowering ketia hobo? Gorom dinot hobo. Ami gorom dinot narakhle na tharante khai fix kori diyo. Right? Jadi if you don't eat the spinach in this winter season and you give the plant like that, the flowering of the plant will take place in the summer. Understood? Apart from these three categories, there are certain other very short SDP, just about 2 hours, that's all. So this is all about photoperiodism. Each and every plant will have their own requirements for day and night length, right? And accordingly, plants are classified as SDP and LDP. What is example? The examples. Second point, in case of SDP, what is important? Continuous dark period. And third point is, increase the darkness in the SDP, good for the plant. Increase the light factor in case of LDP, good for the plant. Next, two terms, florigen, hormone. What is florigen? Florigen is supposed to be a flowering hormone. But whenever we talk about florigen to come equals that there was a scientist called as Khan. According to him, what he said is, we know each and every change in our body is due to hormones. Similarly, if a plant needs to flower, hormones. And so what he said, the hormone that is responsible for flowering is called as florigen. But this is a hypothetical hormone. Hypothetical hormone means, I mean, fact to proof right? But he is of the opinion that florigen is responsible for flowering. So if the question is asked, identify the flowering hormone, what will you write? Florigen. So flowering hormone is florigen. Okay, discovered by Khan, hypothetical hormone. Next we have a word called as phytochrome. Phytochrome, I think I have already spoken at least one line about it. When I was talking about cytochrome, I said we have two words, no? About phytochrome and about cytochrome. What is cytochrome? Electron acceptor proteins, right? And what is phytochrome? Light acceptor protein molecules. These are called as phytochrome. So phytochrome equal to light absorber. And in terms of flowering, no, we are talking about light PAR. Obviously PAR, which light is important? Red light. Right? Okay. So phytochrome discovered by who discovered? Question again. Name of the science is important. So it is the light sensitive pigment responsible for flowering. The term phytochrome was given by Butler. Structure, not very important. Open tetrapyrrole, I think we understand. Closed tetrapyrrole and open tetrapyrrole. Next is, whenever we talk about this sensitive pigment or phytochrome, adult plant of light accepting pigment could be of two types. Either it is PR or it is PFR. Phytochrome ki hoy? Eta protein. Hey, protein to ki loy? Light loy. Ki light loy? Either we can take the red light or we can take the far red light. If it takes the red light, it is called as phytochrome red or phytochrome R. If it takes the far red light wavelength, it is called as phytochrome PFR. So in a plant, phytochrome exists in two forms, PR form and PFR form. Clear? Out of these two forms, the question that is asked is, which one is physiologically active? Obviously, phytochrome far red or PFR form is active, or PR form is supposed to be inactive. Why? Because PFR or far red has a major role in flowering. Red of far red is flowering initiate correct. Right? What we have studied, we have talked about a word called as phytochrome. What is phytochrome? A protein molecule that they will write. How many types of phytochromes or in which forms are they present in a plant? They are present in two forms, PR form and PFR form. PR is physiologically inactive, PFR is physiologically active. Why PR? Because it absorbs the wavelength of red light. Why PFR? Because it absorbs wavelength more than 700. Obviously above 700 is far red, right? Okay. Next is another point is plant out ki thake, PR thake, PFR thake, and both of them are interconvertible. PR to kihole convert hobo pare? PFR le. PFR kihole convert hobo le? PR. If you are talking about PR gets converted to PFR. Red light to kihole convert hobo se? Far red le. Ketia convert hobo? During the daytime when the sunlight is there. PR gets converted to PFR. PFR will get converted to PR during the night. Sunlight night, light factor komi jabo, far red night kya hobo red hole jabo. So phytochrome are present in two forms, PR and PFR forms in a plant. They are interconvertible. PR gets converted to PFR during the daytime. PFR gets converted to PR during the night time. And and which one is active and which one is inactive in terms of growth and flowering. Diagram to this one next page. PR and PFR. Upon chart differentiating PR and PFR, right? If you talk about PR, what is the color it has to be associated with? It's the blue color, right? Taking look at blue onwards, red onwards, that is PFR or the yellow zone. Orange, yellow, red. If you divide the entire wavelength into two parts, one side pura blue, one side yellow. So blue section में जहाँ तक red नहीं आएगा वो सब blue, green, blue, this, that, everything is considered as a blue zone. And once red is achieved, from that onwards is considered as a yellow zone. Diagram understood? PR into PFR? Okay. At a point दिया से a point very important. In SDP, just read that line. In SDP, whether flowering will be promoted or inhibited depends upon the exposure given in the last when two types of radiations are given successively. यहाँ पे क्या है? Example दिया है. Example here of a particular SDP, right? We have taken a plant, key plant SDP, and আমি expose that কি কৰিছো simultaneously, right? So এতিয়া এই প্লান্টটালে flowering সোনকালে কৰিব নে দেৰিকে কৰিব depends on what? Depends on last of কি দিছা. We are talking about this plant. In the first step, what we have taken, this plant, we have exposed it to R. Next, maybe far red. Again we have given red. Again we have given far red in one situation. Second situation, maybe we have taken the same plant. First we have given far red. Next we have given red. Again far red. And then last we have given red. Third of Akko will continue. Maybe a longer period. Right? What you have to see is last of key and say. Last of the R inhibits flowering. Last of the FR exposed korisa to both flowering. Now read the line. If you are talking about a SDP plant and this SDP plant has been exposed to the two wavelengths simultaneously, a plant flowering ketia hobo. R korile hobo ne PFR hole hobo. It will depend on last of key disa. Last of key is the plant is exposed at the last with FR. If the plant is exposed in FR at the last time, then it will initiate flowering. If the plant is exposed to red light at the last, then it will inhibit flowering. So everything depends on what? Everything depends on this last. If you are going to plant R plus FR plus R plus FR and you just go on. R plus FR plus maybe R plus FR and go on. If FR is the last, what will conclude? Flowering hobo no mai. Flowering hobo, right? Arjani, if R is the last, then the flowering hobo. That's his focus. Right? So you both the treatments are given together, far red and red. The flowering depends on the last treatment. And finally, PFR form is biologically active. It helps receive germination, photoperiodicity, etc. etc. To that question we asked is what? Active form of phytochrome. Answer key PFR. Next question is, could a defoliated plant respond to photoperiodic cycle? Defoliated means where the leaves are removed. If you take a plant and remove all the leaves, right, will photoperiodism be observed? No. Why? Because photoperiodic, that photons are taken in by the leaves. If the plant does not have leaves, then the photoperiod is meaningless visible. Right? So this is all about photoperiodism. Photoperiodism Who has discovered what are the different types of plants, SDP, LDP and they neutral. Examples. Next we have to talk about phytochrome. Phytochrome kiman forms or butaform or PR and PFR interconvertible. SDP plant tort. Flowering hobone no depends on what? Depends on the last treatment. Okay. Next, flowering hormone also we have studied. It is florigen. The next unit talks about another phenomenon that is called as vernalization. Vernalization means there are certain plants which always require our exposure of cold climate to flower. এনেকুৱা কিছুমান প্লান্ট আছে যোনে নেকি ঠাণ্ডা দিন যদি পাৰ হৈ নাযায় তেতিয়াহ'লে ই ফ্লাৱাৰিঙে নকৰে. Certain plants which always needs to get exposed in a winter climate to flower. That is what is called as vernalization. Exposure of a plant to cold climate or cold treatment is called as vernalization. Right? So which type of plants generally we talk about is the biennials. Biennials plant, for example, carrot or radish hop. No? I think I have given this example before also. Whenever we talk about such type of vegetables, maybe carrot, radish, etc. If you are comparing two places, Guwahati and Shillong, basically good farm. Shillong, Kibata Thakiboli, Thunder, Thunder key hoy, flowering to easy hoy, flowering to early hoy, more good. Right? So that requirement for that cold climate is called as vernalization. Who is responsible? A particular hormone called as vernalin. Right? Okay. So what I have said, there are certain plants who always require a cold climate to flower. Right? That is what is called as vernalization. Because next part of the lesson will be talking about hormones, right? So one point you have to remember. Suppose we are talking about a plant. A plant to ki lagibo. Maybe you have sown this plant in summer. A plant ka summer opra ki hobo. It will go to winter. From winter, it will come back to summer. Flowering ketia hobo. Etu summer of ki korisa. You have just sowed the seeds, right? Flowering ketia hobo. क्यों? कारण एक winter phase की पाबो लगी बोलते हैं flowering हो. यदि winter phase ना पाए तो है flowering हो, right? So ये process तो कमी की को vernalization. So who is responsible? A hormone that is called as vernalin. One point you may remember is in such type of plants who require the winter time to flower, a good phase to digolia हो जाता है. It is such a lengthy process. Yes or no? If you suppose this is a radish. If you want to have the seeds of the radish, you have to wait such a long time. Instead, what you can do is, such type of plants, if exposed with or if given gibberellin hormone, তেতিয়াহ'লে তাহাঁতৰ winter period একটু নালাগে. Normal case of summer মিলিলা, winter যাব লাগিব, next summer flower কৰিব. This is a lengthy process. You want the flowering to occur early. So what you will do? You will party, direct outsider of hormones. As it is, we know by application of hormones, various things has been done, starting from ripening of a fruit to increasing the size of a fruit, the sweetness of a fruit. So most to keep hormones. Apple is mitha ki ho karne ki man to normally mitha ho nore no. We have applied the sweetening hormone from outside and made the fruit very sweet. Right? Similarly, the grapes nowadays, they are such big grapes. Why it is so big? It cannot be so big. We have applied hormone from outside. So what I have said is, in those plants who require the phenomenon of vernalization for flowering, this winter period, without exposing itself to the winter period, if you want the plant to flower, what you can do? You can apply gibberellin. Gibberellin ki koribo he winter requirement to naikya koribo. Here. Next is devernalization, the opposite. Yeah. Vernalization on the term was coined by Lysenko, you need to remember. Effect of low temperature on the initiation and development of flower is called as vernalization. This effect is, or which part of the plant body takes the stimuli? It is the embryo tip, the shoot apex and the leaves. Photoperiodism boli kole photons bilak kune loy, leaves, right? Flourishing hormone kot thakibo, flowering hormone kot thakibo, shoot tip. See, the flowers are born in the tip of a shoot. So flourishing kot thakibo, shoot tip. If we are talking about this vernalization process, in cold temperature is a plant cell or which part of the plant will take? It is the embryo tip, the shoot tip and the leaf apex. Heibilakor through rate thanda distributed by the plant. Right? Oh, artificial is maybe you can do it. Greenhouse will be controlled atmosphere or the hormone related with vernalization is vernalizing. Next we have devernalization and importance of vernalization. Just see if you have any doubts from this part. Next we'll move to hormones. PRPFR. Plants কিছুমান proteins থাকে, সেই proteins বিলাকে light লয়, সেই proteins বিলাক আমি কি বুলি কৈছো phytochromic. Planter ভিতৰত বহুত proteins থাকে, right? কিছুমান proteins এ electron acceptor, carrier, this that হ'ব, তাৰত আমি কি কওঁ cytochrome, right? যোনবিলাক proteins এ light ল'ব, light ল'ব মানে কি wavelength of light ল'ব, তাক আমি কি বুলি কওঁ phytochrome. So planter এৰ ভিতৰত phytochrome আছে, phytochrome দুটা ফৰ্মত থাকে। দুটা ফর্ম মানে তাতে রেড লাইট টু এবজর্ব কৰিব পাৰে নহলে ফার রেড টু এবজর্ব কৰিব পাৰে যদি প্রোটিনটোৱে রেড লাইট এবজর্ব কৰিছে তাক আমি কি বুলি কম পিআর যদি ফার রেড এবজর্ব কৰিছে তাক আমি কি বুলি কম পিএফআর সো প্ল্যান্টডালত পিআর আছে নে পিএফআর আছে এটা আছে পিআর টু পিএফআর লে কনভার্ট হৈছে পিএফআৰ টু পিআৰ লে কনভাৰ্ট হৈছে পিআৰ টু পিএফআৰ লে কেতিয়া কনভাৰ্ট হ'ব যেতিয়া সানলাইট পৰিব প্ল্যান্টত দুইটা আছে राति की हो पीएफआर टू पीआर हो যাব দিনত কি হ'ব पीआर टू पीएफआर हो যাব মানে একেটাই প্ৰোটিনতে দিনত একদম ৰাতিপুৱা ৰেড লাইট হৈ আছে যেটা সানলাইট কৰিলে বেছিকে লাইট পালে না ই ফাৰ ৰেড হোৱা আৰম্ভ কৰিব আকৌ ৰাতি হ'ল সানলাইট নাইকিয়া হৈ গ'ল पीएफआर হৈ আছিল কি কিহতে কনভাৰ্ট কৰিলে पीआर লে সো দুটা ফৰ্মত আমি ফাইটোটো পাম ঠিক আছে আৰু কিবা ডাউটছ Yeah. So this was the first part. Second part is the growth regulators. The term hormone was given by Purimadise. When we talk about hormones, there are divided into two types: regulators and inhibitors. Right? Regulators mean the hormones that will have a positive impact, will help in the growth of the organism or the plant, that is called as a regulator. If you talk about an inhibitor or negative effects, right? Falling of the part from the plant body, negative, right? So teluka negative effects because inhibits growth, they are called as inhibitor, best inhibitor, abscisic acid. If you are talking about the phytohormones, 5 types of phytohormones, we are talking about auxin, gibberellin, cytokinin, ethylene and abscisic acid. Out of this 5 regulators, auxin, gibberellin and cytokinin, they are regulators. If you are talking about inhibitors, abscisic acid is the inhibitor. Then what about ethylene? Ethylene has both effects. It could be a promoter, it could be an inhibitor also. Etiaba kisuman process of inhibit kore, kintu ripening promote kore. So he has both the effect. Next is the first hormone, auxin. Okay. Now, hormone section on ki physiological role of auxin or physiological role of gibberellin. Right? So these are things that we study. Note to halke poriba. Right? Sentence like each and every sentence that is given is something that you have to read. Right? Because any question could be asked from this part. Taru for you, one statement about each hormone. Auxin. What is the most important effect of auxin? Elongation. Elongation ki hor root tip and shoot tip. Auxin it will have many functions. Apart from them, the most important function is elongation of the shoot tip and the root tip. Second point about auxin, both pi, root apex and shoot apex. Oh. Coming to gibberellin. Gibberellin has many functions. Most important, internodal elongation. You have a plant, you have a stem. Stem of nodes or internodes থাকিব. Gibberellin se dili dia teতিয়াhle ki hobo internodal elongation. Internodal elongation hole ki hobo, the height will increase, right? Gibberellin aru eta point, it acts as anti-dwarfism. Anti-dwarfism. You have a dwarf plant. Hey plant lo ukho koribo bichariছা. Ki diba gibberellin. You have a cabbage. Cabbage ki hoy stem ase leaf ase. It's a compact rosette structure, no? If you apply gibberellin from outside in a cabbage, what will happen? Compact will take cabbage to ekdom dikoliya hoy patbor sob separate hoye jabo. Are you understanding? Because internodes will have compact hoye ase kane rose type hoye ase. Internodes will separate hoye jabo. Leaves will ukho hoye jabo na jay. So anti-dwarfism kar kam gibberellin. Human beings of gibberellin ki bahloli. I will just support and we are like gibberellin from outside and then we become 12. It's only plant. I don't know, all those commercials. However, in that case, I will be happy with that. You don't worry about basically column line. No point. As it is, it's human tendency and nature not to like its own, no? I don't like my height, you don't like your height. Everyone likes the other person's everything, no? He's better, I'm not good. Doesn't matter. Okay, so I was talking about auxin. Auxin ki mona rakiba? Elongation, ki shoot tip, root tip. Gibberellin ki mona rakiba? Internodal elongation, anti-dwarfism. Right? We have a term called as bolting also. Bolt, B-O-L-T. Next, cytokinin. The first line about cytokinin helps in what? Cell division. Cytokinesis. Help for it. Cytokinin help for it. Right? That's the third line. Next is we have what? Ethylene. Ethylene the first line, ripening hormone. The last one is abscisic acid. Abscisic acid inhibitory hormone leads to abscission. Bas. And the auxin or part to the auxin part. Who has given the term auxin? Went important. Chemical characterization of auxin. Who is associated? Mane formula two ne disil auxin or covalent. Right? Next is three types of auxin. No? Auxin A, auxin B are heteroauxin. Auxin A isolated form. Scientists were doing experiments and then they could isolate different types of auxins from different types of plants and they gave different names. Right? The auxin that was extracted from human urine. Auxin A, right? The auxin that was extracted from corn endosperm, that is auxin B, right? And heteroauxin isolated from human urine, which is also known as indole 3 acetic acid. Auxin or good example as a indole acetic acid, indole butyric acid, indole 3 acetic acid, right? So examples ke liye hum जानने देंगे. Next is Went isolated auxin from the coleoptile tip of Avena sativa. What is auxin? Hormone. Who discovered it? Went. In which plant? Avena sativa. What is Avena sativa? Oat. Oats how many? The oats or plant scientific name ki Avena sativa. Right? Next, he stated that auxin concentration in non-illuminated side is 57% and towards luminated side is 27% because 16% of oxygen is destroyed by light. From all these three statements, কি মনত ৰাখিব লাগিব? Just one statement. কি মনত ৰাখিব লাগিব? যে যোনটো সাইডৰে লাইট পৰে তাত অক্সিজেনটো ভাঙি যায়। Oxygen gets destroyed in presence of light. So ইয়াতে কি কৈছে? If you are talking about a plant, right? You are talking about a plant. You have the sunlight falling from this area, right? আৰু তেতিয়াহলে কি হ'ব? এই প্লান্টডাল এপিনে বেন্ড কৰিব নকৰে, right? বেন্ড কৰিব লাগিলে ইয়াতে কিবা এটা হেঁচা মাৰিব নালাগে। সো ইয়াতে কিহৰ হেঁচা? Oxygen develops more কোনটো সাইডে? ডার্ক সাইড। ইয়াতে কি হ'ব? লেস। সো হেতুৰ ওপৰত কোৱেশ্চন আহে, right? কোনটো সাইডে বেছি হ'ব? লাইট সাইড নে ডার্ক সাইড? ইট ইজ অলৱেজ ইন দ্য ডার্ক সাইড। জাস্ট ৰিড দ্য লাইনস। সো বুজি পাবা নাই? Oxygen concentration in non-illuminated side is always more as compared to the illuminated side. Clear? Next is oxygen or aru kelaman question ahe. You have to read something called as precursor of oxygen. Very commonly asked question. What is precursor? The raw material, right? So oxygen is made from tryptophan, right? So remember tryptophan. And for its formation, kuntu mineral lage zinc. Ekini portion very important. So the word precursor is important. Next you have the word called as biosynthesis of oxygen. Biosynthesis mane the oxygen hormone to kot paba. In which part of the plant will you see oxygen? Root apex, shoot apex. That is biosynthesis, where it is synthesized. Next is transportation. If it is synthesized at the root apex and shoot apex, we know hormones bila ki hoy. Jod synthesis hoy, tarpa beleg jagale jai boli, right? So if you are talking about transportation, it is polar, body jabo, tolle walibo, basically, right? Next forms of oxygen, we have natural oxygens and synthetic oxygens. Natural oxygens and synthetic difference we know. If it's man-made, it is called as synthetic. Normally that is available, that is called as natural. Give certain examples of natural oxygen, certain examples of synthetic oxygen. Examples are given. Natural oxygen we call it indole acetic acid, very very important, right? And indole butyric acid also. Next we have free oxygen and bound oxygen. If the oxygen or if the hormone remains associated with something else, it is called as bound. For example, IAA, indole acetic acid, is an example of free oxygen. IAA dash aspartic acid, aspartic acid, that is an example of a bound oxygen, right? So oxygen or examples kini saba. Next, antioxidant. Kisuman hormones, kisuman chemicals. Junbilak apply korile oxygen or kamto naikia korilibo. That is what is called as antioxidant. Next, we have physiological effects of oxygen. Coming to physiological effects of oxygen, read the first point. Oxygen is necessary for cell elongation and longitudinal growth. It will increase the length, no? Next, oxygens are present in sufficient amount in the growing tips. Shoot tip and root tip is present. So, exogenous oxygen application given to intact plants has no effect. This is a very important line. Okay, what we are talking about is hormones. Whenever we are talking about this plant hormones, we know these are the hormones that are already present in the plant. But in spite of that, we have made some synthetic hormones also. Synthetic oxygen one. For economic benefit, right? So indole acetic acid is there, but like that something else we have paid, right? So whenever we are talking about artificial hormones we apply it to the plants to give good results, right? So if you are talking about synthetic gibberellin, you can give that chemical to a plant. If you are talking about synthetic auxin, you can give that chemical to a plant also, right? But one thing that you have to remember is, just take two conditions. You have a plant here, you have a plant here. In this, you are giving him synthetic auxin, right? You have a plant. Similarly, you have a plant here also, right? It is a synthetic gibberellin. Right? The point is, synthetic auxin to modify the result. You want something good, you want to increase the height, or maybe, right? So if you have to give synthetic auxin to a plant, this plant should be if this synthetic auxin needs to work in the plant, a plant that intact hobo nalagibo. Now what is the intact plant? If you don't remove any part, that plant is called as an intact plant. If you are talking about auxin for thakibo, maximum yat thakibo. So if you want to apply some type of a chemical similar to auxin to this plant, you always have to remove the tip. This is what is called as non-intact plant. If you don't remove intact plant, if you remove the source of natural auxin, that is a non-intact plant. So if you have to apply hormones from outside, especially which hormone? Auxin, then you always have to remove the original source of auxin. Root tip to তুমি shoot tip to গুছাব লাগিব, তেতিয়াহে বাহিৰৰ হৰমনটোৱে কাম দিব। যদি গুছাই নিদিয়াকে বাহিৰত দিয়া, তেতিয়াহলে কাম নিদিয়ে। বুজি পাইছে? কিহৰ কাৰণে? কাৰণ it has enough in its tip. You have to remove the natural source, then only the artificial will work. How will you remove the natural source? By removing its tip. Why the tip? Because auxin is present in huge amount only in the tips. Which tip? Shoot tip and root tip. Right? But in case of gibberellin, you can apply synthetic gibberellin in non-intact plants also. Auxin, if you want to apply, key plant non-intact. Sorry, it should be. Sorry. Right? Auxin যদি বাহিৰৰ পৰা দিয়া, তেতিয়া প্লান্টডাল কি হ'ব লাগিব? Non-intact. Gibberellin যদি বাহিৰৰ পৰা দিয়া, প্লান্টডাল কি হ'ব লাগিব? Intact. So it does not matter. But if you have to apply auxin from outside, you always have to remove its tip. And that removing of the tip of a plant, that plant is called as the non-intact plant. Now read that line. Exogenous means application from outside. Is the line understood? It will not question I. Next, ki diya ase auxin is one role. This physiological role you have to read. Just read any point you did not understand, ask me. Auxin is also helped in cell division in the cambium, cell division for tissue culture, etc. etc. Important point number 4. Low concentration of auxin, he developed for a phloem. High concentration of auxin, both silent and phloems developed. Point number 5, important. Low concentration of auxin promotes root growth. High concentration of auxin inhibits root growth. Point number 6, the word is very important, apical dominance. What we understand by the word apical dominance? Apical is related with apex. Dominance means, okay, just tell me, if you want to make a plant bushy, what will you do? Kisman plants, for example, Tuloki Kausal dekhiso no ami. Tuloki Kausal whenever it grows, no, the If you want to make it bushy, what you do? The second example could be tea also. You have seen a tea plant, right? So the tea plants are really what? It's like a bush, right? So what do you have to do to make a plant bushy? Or how does the bushy nature develops? If you are talking about a plant, the plant will always grow by the radical and the plumen, right? It will grow slowly. What you want to do, you want to see that lateral roots arises. The point to be remembered is, if you want these lateral roots to arise, you have to remove the tip. Right? Tip remove korle ki hobo? Oxygen naiki hoy oxygen howa kole elongation no hoy, growth will take part through the lateral side. Again, in this tip, ki asile oxygen, you remove this tip. Right? Again, this was a branch. In this tip, ki asile oxygen, remove this. Right? So ultimately, oxygen se bhi thake, tetiyale ki hobo? The apex will keep on growing, and this is what is called as apical dominance. Who is responsible for apical dominance? The hormone oxygen. To remove apical dominance, what you need to remember do? You have to prune the tip. Next, Makita point for some prevention of abscission layer. What is abscission? Cut off. If any part of the plant body needs to fall off from the plant, tetiyale ki hobo? Adult plant ase. Plant will suppose a fruit ase. Right? This fruit, once it has ripened, it will fall down. Before falling, this falling or cutting off is called as abscission. Before it falls, at this point, there are certain cells that will develop, and this zone of cells is called as abscission zone. Right? There should be certain cells, they should form a zone. After the formation of the zone, the fruit will fall down. So if any part of the plant needs to fall down, by abscission hobo lagile, ki form koribo lagibo? Abscission zone to form koribo lagibo. Abscission zone form koribole kune help kore? It's a negative part. Who helps? Abscisic acid. But you have a mango plant in your house. ठीक है सर? Mango plant so poki gol. Mango poki joar pisot ki hobo? Kori koribo nokore. But you want the mangoes to stay in the plant for a longer duration. You don't want all the fruits to fall off. What will you do? You will apply hormone. Which hormone? Oxygen. What will oxygen do? Stop preventing this abscission layer. Is the point understood? Normally, gosala ase gosot am lagi ase. At a particular time, on ambur kori koribo. Right? But we want am kina aru kelinman thako, aru soma thako. Ki koriba? You have to do something, no? Obviously, God has given us the brain, so we apply in a negative way, positive way, both ways, right? So what we will do? We'll play with the plant now. How will we play? We'll apply hormones. One hormone that we have understood is oxygen. What do you do? You'll take the plant, remove the root, tip, apply oxygen from outside. Oxygen dile ki hobo? Abscission layer form no koribo. Jodi abscission layer form no kore, abscission no hobo, the fruits will not fall down. So good, na? And so bad also. Obviously, everything is artificial, it's never good. Is the point understood now? So now read point number 4. No, point number 5, 6, 7. 7 point. Is point number 7 understood? Understood now? Delays the abscission of young leaves and fruits and promotes the abscission of. Okay, it promotes the abscission of mature leaves. Yeah, at a fruit, young stage of kori no kore, mature hole kori kore. Who plays with all these things? Hormones. Which hormone? Here we understood oxygen. Oxygen will never allow a younger fruit or a young leaf to fall down. Right? It will allow the mature one to fall down. Okay, understood. Next is point number 8. Parthenocarpy. What is parthenocarpy? Understood now? Without seeds, without fertilization, etc. etc. So parthenocarpy and the role of hormones, important. Different hormones are responsible for bringing about parthenocarpy in different plants. Question ahe, yate eta example ase tomato. Direct question ki ahe se, who brings about parthenocarpy in tomatoes? Once we'll be reading about gibberellin, we will see some examples of plants which will undergo parthenocarpy because of gibberellins. Kisman plant enege ase parthenocarpy because of auxin. So that example oriented you need to study. Udipo tomato parthenocarpy kune aaye auxin, right? That example very important. Next is flower initiation. We understand. Point number 9, very important. Auxin or gudegida point important. Please read point number 9, flower initiation. Flower initiation point to padha. Once you have read the point, if I ask, what is the role of auxin and flowering? What is the role of auxin and have you read point number 9? Read? Okay, now answer me. What is the role of auxin and flowering? Okay, if I put it other way around, auxin inhibits flowering, then auxin promotes flowering. Inhibits flowering. So if in a plant auxin concentration is more, then the plant will not flower easily. Obviously, because it's responsible for the vegetative part, no? Shoot apex, root apex, right? So auxin basically is flowering normal. Kintu you have an exception, and the question is asked about exception only. What is the exception? So some pineapple, right? So that is the way you read. So question ahead: who initiates flowering in pineapple and lychee? Auxin. So normally, what is the role of auxin in flowering? It inhibits flowering. But in case of pineapple and lychee, it promotes flowering. Respiration. Uses of auxin. Next page, very important. Right? One of the most important function of auxin. Auxin is used to bring rootings on stem cuttings. Stem cuttings, cutting, layering, all this we have studied, right? So stem cutting is another class. For example, bougainvillea, or you are talking about some other plant like rose. You want to grow another rose plant. What will you take? You take a stem, piece of a stem, right? That piece of a stem could be planted in the soil, and after a few, maybe time or maybe some months, you will see a new plant. But if you want to hasten the process, what you can do? Take the stem cutting, apply the hormone, the roots will develop, plant it down. Right? So, rooting on stem cuttings. Hey, what is animal? Who is responsible? Indole acetic acid or auxin? Or indole butyric acid? Or NAA. NAA is full form the acid, naphthalene acetic acid. Okay, identify the BD sites. 2,4-D or 2,4,5-T. These are certain examples of auxins that are used as BD sites. They are also called as agent orange. Question is identify agent orange. What is agent orange? Agent phenotype, no? So agent orange ki bostu. Agent orange ko duta BD site. Right? Kuntu hormone auxin. Right? 2,4-D or 2,4,5-T. Point number 3, again important. Methyl ester of NAA prevents sprouting of potato tubers kept in storage. Again coming to the example of potato. If you bring a lot of potatoes in storage at your home, after a few days, what will you see? Sprouting. Gozali melibo nemile. Kinte ekekita potato, if it is in the store, dukanot thakile gozali nemile. Kyo? Hormones. Auxin. What do? They apply those chemicals. As a result, what happens is it prevents sprouting. So question ahead: Who prevents sprouting of potato tubers in storage? It is so hormones will be compare korim na. All of this ami eta hormone pam who will help in sprouting. A2 hormone ki inhibits sprouting. So that's the way you have to read it. Okay? So question number 1, 2, 3. Bute kita ahe.
Point number 2 name 24D.
24D. Full form ase na swa. 24D is 2,4-dichlorophenoxyacetic acid. Page number 70. Page number 81. Is it 83? Synthetic oxygen examples what? Synthetic oxygen page number 83. 82 or 83. Synthetic oxygen examples what? 2,4-D. Alright. So 2,4-D, 2,4,5-T. These are synthetic oxygen that are used as weedicides. Next is pre-harvest fruit drop. Pre-harvest fruit drop. Khori jate nokore. Khorute etia harvest ketia hobo jete hi mature hobo. Mature nohua ke jate hi khori nokore. Gosdalot ami ki dibo paru hormones dibo paru. Right? So it's pre-harvest fruit. It will prevent pre-harvest fruit drop in case of orange and apple, and also in case of tomatoes. Generally, if you have seen a tomato plant in your home, tomatoes have a very high tendency to fall off. As a tomatoes but tomato so it's a very negative, it brings a negative impact to the farmers. So what does the farmer do? A plant hormone that is what is called as pre-harvest fruit drop. Very bad. Very obviously negative impact. All these chemicals will have a very bad impact on you. That is the reason why so many new diseases are coming up nowadays.
Ma'am, point number 2, ma'am, Agent Orange is
Agent Orange is a name given to the weedicides. Right? Agent of weedicide. Oxygen আছে সেই oxygen কিবা কি হিসাবে ইউজ কৰিছে weedicide হিসাবে ইউজ কৰিছে সেই weedicide এটা এটা নাম দিছিলে Agent Orange. Okay. Now the last point that is important is bioassay of oxygen. If you have gone through the MCQs, this is a very common question that is asked. What is the bioassay of oxygen? What is the bioassay of gibberellin? By the word bioassay, what do you understand? The experimental course. Right? Bioassay of oxygen, experimental course. Oxygen discover experimental course. Oat. Right? So bioassay of oxygen is Avena curvature test. Avena ki asle oat plant. Right? So wherever you see this word Avena or maybe coleoptile, we understand that this is bioassay. Bioassay ka ami mukhast kori. Oxygen or bioassay, gibberellin or bioassay, etc. Right? The experiments. Next is Claude-Lewin theory. Alright. Claude-Lewin theory talks about what? More oxygen is synthesized always on the darker side. That is what is Claude-Lewin. And a question we asked the probability what? Bioassay of oxygen is. Bioassay of oxygen is the Avena. What is bioassay? The experiment that was conducted. Right? Second question that is important. Sprouting of potato tubers under storage could be prevented by oxygen. So this is all about oxygen. Just go through oxygen. If you have any doubts, please ask. So oxygen or in a lump sum, what we have studied? First, shoot tip and root tip. Second, apical dominance. Third, physiological roles, right? Parthenocarpy code, VD-sites or NAMKI, right? Next is pre-harvest food drop, etc. etc. Fourth is more oxygen on the light side than darker side, obviously on the darker side, right? That is fourth point. Fifth, human urine and oxygen. Sixth, natural oxygen, artificial oxygen, some example. Sixth point, rooting on stem cuttings, very important, right? Seventh point, bioassay. Eighth point, potato tuber. That's all. Stop. Go to the oxygen portion. Any question, please ask. Cloudiment theory states that unilateral light produces more oxygen and hence more growth in the shaded side, resulting in bending. Tarmane, Cloudiment ki eta word use korisile unilateral light. Koise no? Unilateral light mane ki? Je tumi suppose edal plant ase na? ঠিক আছে এই প্ল্যানডাল তুমি এটা বক্সত থৈছা আৰু এই বক্সত এটা ফুটাৰে লাইট আহিছে একেলগে বুজি পাইছা এটা প্ল্যান্ট আছে প্ল্যান্টটো বক্স এটা থৈছা বক্সত এটা ফুটা আছে এই লাইটটো অকল এটো সাইডে আহিছে এটো আমি কি বুলি কম ইউনিলেটাৰেল light coming from just one source, right? So whenever light comes from this source, a plant dale ki koribo a light or ipinole bend koribo nokore junpine light ahise epine kotte straight asile junpine light ahise costal bend koribo no, right? So Clodney bend ki korisile je etu kihor karone hoise এই যে বেন্ড কৰে যিকোনো এটা স্ট্রেট বস্তু বেন্ড কৰিব লাগিলে এটো সাইডে প্ৰেসাৰ কৰিব লাগিব না লাগে সো ইয়াতে গটে দিছে যে এই প্ল্যানডালৰ এটো সাইডে যে বেন্ড কৰিলে এটো সাইডে গ্ৰোথ বেছিকে হৈছে more growth এটো সাইডে যে more growth হৈছে তেতিয়াহলে প্ল্যান্টৰ গ্ৰাজুয়ালি এপিনে বেন্ড কৰিব নকৰে কৰিব সো এই more growth কিহৰ কাৰণে হৈছে কাৰণ এটো সাইডে যত নেকি লাইট নাই তাত কি হৈছে more oxygen, right? Oxygen ক'ত থাকে টিপত থাকে, right? shoot tips root tipot oxygen থাকে যোনটো সাইড লাইটৰ পৰা দূৰত আছে সেই সাইডটো বেছিকে অক্সিজেন ডেভেলপ কৰিব বেছিকে অক্সিজেন ডেভেলপ কৰিলে বেছিকে গ্ৰোথ হ'ব বেছিকে গ্ৰোথ হোৱাৰ কাৰণে গছডাল বেন্ড হৈ যাব টুৱাৰ্ডছ লাইট দিস ইজ Clodney bend theory clear
respiration se kya oxygen कैसे respiration में होता है
which one
respiration
oxygen induces the synthesis of various enzymes वो वाला point helps in the formation hormones are responsible for each and every function right so if oxygen is more certain types of enzymes responsible for respiration will increase as a result more respiration bioassay is a term just simply ki mono rakiba যে it talks about experiment. কিছুমান এক্সপেৰিমেন্ট কৰাৰ পিছতহে হেৰি হৈছিল যে তোমাৰ অক্সিজেন যে এভিনা পলিঅক্টাইল টেষ্ট কৰিছে ন এটা প্ল্যান্ট হৈছে প্ল্যান্টৰ টিপটোত যে অক্সিজেন আছে এইটো এক্সপেৰিমেন্ট হয় ন এক্সপেৰিমেন্ট হয় তো সেই এক্সপেৰিমেন্টটোৰ নাম আমি কি কৈছো বায়োএছে সো বায়োএছে অফ অক্সিজেন বুলি কোৱেশ্চন আহিলে উঠে কি যে অক্সিজেনৰ লগত ৰিলেটেড কোনটো এক্সপেৰিমেন্ট হয় ডিসকভাৰ কৰিব লাগিলে কোনটো এক্সপেৰিমেন্ট কৰিছিল আমি এভিনা পলিঅক্টাইল টেষ্ট কৰিছিল Aruswa, anything about oxygen? Apical dominance. Apical dominance matlab if you are talking about a plant, right? This is the apex. This apex will keep on growing. If this apex keeps on growing, lateral growth will not take place. This is called as apical dominance. Why it is dominant? Because it has oxygen here. If you remove this source, oxygen is absent. Oxygen is absent. ये part और grow नहीं करेगा, right? Then the growth will start from this part, right? So who is responsible for apical dominance? Oxygen. Elongation of the apex. It could be shoot apex, it could be root apex also. Once you are done with oxygen, please say. Morphological features according to light. Light has a role in the growth, no? The light korile or light nokorile, common to morphology change hobo, no? That is what is called as Would you oxygen give a police to any point that you did not understand? Please ask. Due to the point important, also please be careful. Uses of oxygen important. Next is that parthenocarpy, flowering, parthenocarpy is that point important. Root. Exam of kuntu exam. Ever responsibility Mukesh is the job MCQs bila koribas news bila ko idea pawa previous year questions aba So should we start with gibberellin today or we'll do it in the next class? Next class? May also not get out of the Yeah, please say, someone say something. Intact or non-intact plant? No. intact plant or non-intact plant. Adult plant আছে, plantor tar fruit apex to আছে, so parts bilak আছে, tak ami ki boli kom intact hoye ase, tamane eku part tumi remove kora nai, tak ami intact kom. Adult plant আছে, jar ei shoot tip to tumi kati belaisa. বুঝি পাইছা? এটো বস্তু কাটি দিলা ন? যদি শুটটিপটো থাকে তেতিয়া তাক আমি ইনটেক্ট, তাৰ মানে একো পাৰ্টে তুমি গুছাই দিয়া নাই বা কাটি দিয়া নাই, কিন্তু এডাল গছৰ তুমি আগটো কাটি দিছা ন, সেইকাৰণে তাক আমি কি বুলি কম? নন ইনটেক্ট। আৰু অক্সিজেন যদি বাহিৰৰ পৰা দিব লাগে, যদি তুমি আৰ্টিফিশিয়াল অক্সিজেন বা সিনথেটিক অক্সিজেন তুমি এপ্লাই কৰিব বিচাৰিছা প্লেন্ট ডালত, তেতিয়া মানে সদায় কোড এপ্লাই কৰিবা? নন ইনটেক্ট প্লেন্টত। synthetic oxygen applied on intact plants, no result. Synthetic oxygen applied on non-intact plant shows result. বুঝি পাইছা? Is this point clear? Intact, non-intact clear is it? Synthetic oxygen copra diba. Outside of it.
ये एसिड का नाम को याद रखना पड़ेगा।
कौन सा?
ये एसिड का इंडोल एसिटिक एसिड।
Endo, not all. 3, 4. IAA, IBA, NAA, 2,4-D, 2,4,5-D. Right? सब कुछ नहीं चाहिए. Generally, these 5 are important. IAA, IBA, NAA, 2,4,5-D, 2,4,5-D. Or maybe another one is dicappa. It should be चाहिए. इतना important नहीं है. These 5 are important. So, are you about doubts or seeing? So, today in the class what we have discussed is we have talked about some points about growth and development, right? We have talked about the different types of growth, relative growth, absolute growth, metric growth and arithmetic growth. What is important? How to calculate relative growth, that is important. Formula of geometric growth, formula of arithmetic growth, that is important. Certain terms called as plasticity, heterophilia, examples of plasticity, examples who shows heterophilia, important. Next, what you understand by cell growth or phases of growth, cell division, cell elongation and cell maturation, that is important. Next is we have talked about photoperiodism. Photoperiodism, who has given the concept in which plant it was experimented upon, SDP example, LDP example. Requirement, most important requirement for SDP, continuous dark period, right? Next is we have talked about what? Phytochrome. Phytochrome, we understand light absorbing or photons absorbing. How many forms? 2 forms. Which one is active? PFR is active. PR into PFR, PFR into PR, interconvertible. During the day, PR to PFR, during the night, PFR to PR, right? Next, in case of SDP, whether which application will initiate flower rate depends on what? The treatment that is given in the last. Any last of the art came, any red light key or stop, right? Anything last art, then flower memorial. This is a way to remember. This is not the correct explanation, obviously. Just to remember, you have last FR and in one condition you have last R. So R means stop. So stop, so no flowering. Just remember this one. Okay. Next is we have talked about virilization. Certain plants, which plants? Generally the biennials. Example of a biennial, carrot, radish, etc. are biennials. What they require? A treatment of cold temperature for what? For flowering. What is the hormone responsible? Vernalin, right? Next, the effect of vernalization could be overcome by which hormone? Gibberellin, right? GA we can apply. So those were the points that we have discussed, and we have started with auxin. Auxin is 5-6 points, that's important. I think that's all for today. In the next class, we will be talking about gibberellin and